I guess we've begun. Welcome to Micro. For most of you, if I don't know somebody, I'm Dr. Stein, and hard stuff for many years. I mean, last time it was one lecture or one lab of science. Now it's typically 2 lectures, 2 labs of science. So I don't even have fun. And so it becomes every step for a lot of you that are taking this particular pathway. I know it's not necessarily everything. I personally think micro is the more difficult of these, of AP1. AP1 is hard because it's the first time you're around. AP2 is really it's more physiological. It's really starting to put the pieces together. Bones, muscles, nerves kind of give you an idea. Now you're going to look at how the heart works, how the lungs work, your urinary system, immune system. So and this ain't easy. The trouble with micro is it's not what you're used to. We sat there, and I basically had everybody in AP1. It's like, okay, here's the cell, and we're used to that cell. The nucleus, the mitochondria, all that stuff. Totally different. We're looking mostly at bacteria and viruses, and they ain't nothing like this. It's a totally different form. We're looking at a different kind of cell called a prokaryotic cell. So we've spent most of our lives studying, most of your life studying is called a eukaryote. It's a major difference. We're going to go over all this. And then we're getting to things that aren't native cells, which are not living by definition. Viruses. My wife and I last night we talked about vaccines and influenza. Oh my God, she got the flu. Oh my goodness. I don't know if you know anybody in Europe that really had the flu hard over this break. Oh my goodness. Down for like 2 weeks. Yes. And they're talking about versus live vaccine. There's no such thing as a live vaccine. They market it that way because viruses aren't alive. They're not made out of cells. You're either living or dead. You're either active or inactive. And we'll get to that. And that's a whole different thing. So all of that's the unique part about microbiology. This micro that you're taking is a little bit different. And this is the micro that's designed for health science, for people just taking micro, maybe even a little bit for environmental science, typically nursing and healthcare oriented careers. Our bio majors take a different micro, which is more centered around microbiological things, more in their anatomy and physiology, and looking at microbiological systems. My focus point is different because I know Dr. Maurer is teaching another section in the afternoon. I'm much more clinically oriented, meaning I spend about the last third of this on diseases. That's what I know the most about. So it's like I try to get through we have to do anatomy and physiology again, except this. Here is what to learn. So all of this is there. There's a sign-in sheet, but now everybody's here, so that's good. I just have to count to 26. Right, and last time 32 and 33. They make a big deal about understand, and with all of your classes, again, we have a big lecture about this in our faculty meeting. It's tied to federal money. We have to do this. We get yelled at a lot. So understand. So we have to do I have to do this until I can kind of get used to who's here and who's not here and figure it out. Once again, I'm recording. I'll do the YouTube thing. If you want to watch them. Most of you only look at the ones that have to do that are pertinent to the review record. It's okay. Okay. That's all so I think you get the mechanics, the idea. This is a very difficult thing that we're embarking on. We're looking at, I never know exactly where I'm going to start. I have a lot of material. Let me show you that first. Okay, syllabus wise, put that thing. You read it. I mean, I didn't change it from last year. Same thing. 4 exams, same mechanics. Homework, your homework, read, study. No extra credit. We have that number. Most of you realize how user-friendly I was last term. I don't want to pat on the back. I understood the difficulties that were presented to a lot of you last term. Because that's last term. Please. There are limits. Because you have to transition because people are not going to be giving you those depending on where you're going in your major field, they're unlikely to be as user-friendly. I understood that it's a difficult transition, and I think a lot of the transition has become more difficult because COVID and remote learning and a lot of the stuff that we do now because we just become accustomed to doing it. I think that has impacted a lot of students compared to the traditional model. That's me. But I'm old fashioned. Shout out my ex. My wife and my ex are both January birthdays. Interesting. And her my wife, her husband was born in May like I am. So my ex is hit the 3/4 bulb today. She's 75, and yours truly, May 10th, with those cards and presents coming, 75. I know, I'll still be here. 3/4 pole. I promised the boss I'd be doing this till I was like 95 or 100. She knows it's gonna happen. She's gonna wheel me in a wheelchair and use the same bad jokes nobody understands, the same bad references no one's ever heard of. Okay, all completely. It's true. Moving on. So, 4 exams, the final, not cumulative. Believe me, you don't want it to be cumulative. There'll be announced exams, study guides, the same type of stuff, except got to be on top of it. This stuff is hard, and it gets hard right away for the first exam. It will tie in with. Does anybody have micro lab yet? Oh, you did. Okay. Well, who was doing the lab yesterday? Dr. Oh, nicest guy in the world. Close to. We'll try to tie this in with the lab. I know the introductory lab is pretty straightforward. But very soon you're going to get into things called gram-positive and gram-negative. The whole idea of bacteria is, that's really what your lab is, what we call a BACT lab. Bacteriology lab, that's what you'll be doing. You'll probably have unknowns and a variety of different things. It's a pretty well constructed lab. And the lab and micro lab is a lot of fun. Micro lab. So that you read the syllabus as much as I can. I'm trying to watch my time so I can get through all this stuff. Did you have any trouble logging on with your package into the textbook? Have you tried? All worked fine. Usual, yeah. I had them do it for me. I know I should be able to know how to do it. So everything else is pretty straightforward. All the different stuff that's there, et cetera, et cetera. So I can do without that slide, can't I? Yes. Which brings us back to the Blackboard. So you'll find the syllabuses on the Blackboard. Where am I going? Correction. Okay. And I loaded these last couple of days when I got busy. Okay. So, and this is the first PowerPoint that you see up there, all there. The textbook is there. The PowerPoints will be there. I have a lot of other resources. For instance, this chapter 1 doc, this is a brief outline. I've got outlines for some of the chapters, not unlike what we saw in AP1. Not all of them, but I have some very useful outlines, particularly early going. Maybe most of the first handful of chapters we are doing, I have some pretty useful outlines for that. And in the later chapters, when we get to the diseases, I have some useful outlines. So, and I think you'll find those. So here you have the PowerPoints for this first sequence. I'm not sure for the first exam how far we're going to get along here. 2 is mostly lab stuff. I won't be using it at all. I put it on there for your convenience. 1, you'll see we're going to start with today. 3 and 4 are really about the bacteria. Really, you're going to be looking mostly at bacteria, their structure and how they work, and maybe some additional other microbial organisms other than bacteria. Chapter 6 is really a bit about their physiology. And I mean, this is really where the crux of the material will come from 1, 3, and 4, and possibly aspects of 6 for this first exam. There's what to do with regard to that. I'll show you the outline for chapter 1. This is a supplement I took from another micro text, which goes into a lot. You'll find that. Plus, it has some lab stuff in there. We'll look at that when we get to that chapter. I look at this. Historical figures are very important because you got to understand how we kind of figured out how these things work. It's very interesting historically. Very, very interesting historically about all of this stuff. And it's going to become very topical. I just mentioned a horrible flu. It wasn't COVID. Okay. So we're going to have to understand the distinction. We'll do a little unit on what COVID is, what misconceptions about it, etc. This is a flu. This is by no means, this was by no means one of these unusual, what you would call swine flu or bird flu type things, but the cause of the nature of what it is, and you're going to read about this was called an H3N2. And we'll understand what H3N2 is and H1N1 is and things like that, because it's very, very relevant for all of those things. Just for instance, so you have to go through we're going to look at the history. I have some additional notes for all of these. Voila, plenty of good stuff that's on there. Anybody snowed so far? Good. All right. I suppose that means we can start. Did I forget anything? Nah. All right, let's look at this. Look at the outlines over here. This for whatever the settings on this computer, they're very small. Now, maybe they're not small to you, but I actually inquired with HR because I have to get my vision checked. I'm just not seeing things the way I would like to. Okay, so this is what we're going to look at. We're going to look at the basis of the microscope. You might have covered that in lab. Did you talk about the basis of the microscope? Binocular, von Leeuwenhoek, that's the historical stuff a little bit. I know you talked about refraction and things like that, right? Bending the light in lab. That's what we don't do it. But we're going to look at all of these things. We'll look at all of this will be covered in the first chapter. As you can see, there's a lot already, and you begin to, and there's a fair amount of nomenclature that goes on as well. We have to name them. Fortunately, I try to keep it relatively simple, and I would guess Dr. Dress or whoever else is teaching the lab would probably do the same thing. So if you're going to, for instance, we use the term E. coli. You've all heard it. Its full name is Escherichia coli. This was named after a doctor who had discovered it named Escher. Coli because it was found in the colon. Okay, nothing magical about it. A lot of times those are acceptable abbreviations. I will comment on that a lot, because many of you are going to be responsible for charting, and charting is really important to get everything exactly the way it's supposed to. Charting is anyone's defense against potential litigation. I mean, you have to be introduced to the medical legal aspects, particularly if you're in a healthcare field. You believe me, you do not want to be on the business end of the lawsuit. I have, unfortunately, a little experience of that. It ain't fun. Okay, so we'll get into all that good stuff. So that's there, which brings us to where we should start. I did that in about 13 minutes. I'm happy with myself. I hate this little box is driving me with sugar. Crazy. Okay. So this is the first PowerPoint. I'm using these. You're going to see I'm going to substitute PowerPoints, and they'll say, and the name will be Nestor, N-E-S-T-E-R. That's the textbook we used to use. I agree that this is, and I've used both of these textbooks many, many times. This is called Cowan. So the Cowan textbook I used in half dozen different schools, and the other schools, and for years we used the Nestor textbook. I like different aspects about both. You're going to see I like the illustrations. I like animations that I'll provide for you. You're going to see things like this. You're going to learn. It's a whole different world when you talk about these things. And it goes back to that same thing with the flu. I did a little research about that on the flu and the flu shots and all like this. Statistically, so far this year, that flu shot has having a success rate of well under 50%. My wife and I didn't get flu shots, by the way. We haven't for the last couple of years because the success rate is going down. Understand that's because you're going to see there's a lot of genetic variations. That's really the most difficult thing to understand, yet the most important thing to understand. We spent a lot of time in A&P talking about our kinds of cells. Our cells are relatively stable. We make billions of cells every day. We make very, very few mutations. We have a lot of genetic safeguards, biological safeguards, chemical safeguards in our system that prevent mutations. And if they occur, our immune system smashes them. Unless there's an idiot who spoke them previously. But no such thing with bacteria or viruses. They mutate all the time. That's the biggest single problem we face in medicine today. Okay? The difficulty with finding antibiotics and antivirals and things like that because we have to treat them. Why didn't the flu shot work? Because the flu is mutating as we speak. The biggest single problem we have with regard to taking care of bacterial infections, we're running out of antibiotics. Not that we don't have enough of them on supply, they don't work because the bacteria are mutating constantly. If I said to you, if I said to you, what's a common infection we see in the hospital? What would you pop out and say? Anybody? They're not great. Pardon me? Flu's viral, but good idea. Okay. She gave it to you. That when I asked you to throw something out there, we didn't have a whole lot of it. I understand that, but this is you learn. You learn when you're right, and you learn when you're not right. That's a good distinction that's there. Has anybody heard the term MRSA? MRSA. You're scared to death of it in the hospital because it's a mess. It's difficult to treat. There are different kinds. It means methicillin-resistant Staphylococcus aureus. The most common infection we have is a staph infection, Staphylococcus aureus. When we first discovered penicillin, I'll reiterate this again, 90% of all penicillin were 90% of all Staphylococcus aureus were destroyed by penicillin. Easy. We have evolved that bacteria times 2. Not only just 90% of them are resistant to penicillin. Now we have the vast majority of what we encounter in the hospital are resistant, and not just resistant to penicillin, they're resistant to what we try to develop in the place of penicillin, which is a cousin of it called methicillin. When we do our unit on antibiotics, I'll go over this in chapter and verse. We're running out of stuff, and it's only getting worse. Only. That's why the relevance of all this is here. But let's begin before I get overly preachy about it. Oh. I'm sorry, I haven't talked to a group of people in 5 weeks, thankfully. Believe me, as you all filtered it, I'm like, oh, am I really want to do this to myself? All right, here we go. What's the they give you these things. Yeah. It's good. Here they are. It deals what is meant by microbiology. Micro means you can't see it with the naked eye. And effectively, not all of these things, most of these things you cannot see with living things that are ordinarily too small to be seen without magnification. That's why the microscope becomes such an important tool that we utilize. Bacteria, effectively, you can't see them. Archaea is something that I will talk about historically. They're a cousin of bacteria, but they only exist in extreme environments. Polar ice caps, inside a volcanic caldera, inside the opening for a volcano, bottom of the ocean. They're being studied scientifically for some of the things that they do. And really, we may find a lot of interesting things from them. They're really not part of our course. From a definitional standpoint, I will go over what's relevant from the course. Effectively, they're not exactly the same as bacteria, though they're very similar, and they live in extreme environments. Bacteria live everywhere. And they're covering your entire body. My body, everybody's body, in its entirety, right this instant. 100% of your skin covered with Staphylococcus and a couple of other microbes. Doesn't matter. You look like most of you showered. It's still there. Believe me, it's still there. Protozoans. Protozoans are the kind of things, they're a little different, as you will see. They're the kind, maybe when you were in high school and you got pond water and you looked at things swimming around and spinning around it under the microscope. Is it a bell for some of you? Okay, those are typically protozoans. Protozoans, they don't require as extreme a degree of microscopy. If you look at bacteria, you have to use oil immersion to identify. It might magnify 1,000 times. These you can see with a standard light microscope without oil, that high power. Okay. They're equally as important. We dismiss things out of hands, and we should not. The second most common sexually transmitted disease is called Trichomonas vaginalis. It is a protozoa. It causes the same kinds of complications as chlamydia, many of the same complications as gonorrhea. The second most common sexually transmitted disease, human papilloma virus. So bacteria aren't even in the top three when you talk about sexually transmitted information. We do a unit on that at the end. So that's protozoans, and there's others that play a role. Fungi, fungal infections, athletes would be a perfect example. Jock itch, that kind of stuff. All those kinds of things are fungal infections. Fungal infections are very significant. Not so much that they're that difficult to treat because we can treat most of them topically. Good news, bad news is they open the door to bacterial infections because they violate barriers. We're going to talk about barriers. Your protection, your number one protection to disease is your skin. I don't do helminths are worms. I say it all the time. If you want to know more about worms and you have a veterinarian for a pet of yours, that's who he is. They deal with that stuff all the time. We don't see a lot of worms because it's associated with water supply when people get them. So as long as you have we have basically a pristine water supply. It doesn't all taste the same, but our water's pretty good. So I mean that's why I don't know enough about it to teach you that. That's why I don't. There's some aspects of the therapy that are interesting that we have uses for. Algae or something very significant worldwide. You're going to see the uses of algae. Algae are things like red tide, which can be very poisonous. Again, I don't know a lot about it, but I'll give you. I mean, this is mostly when we introduce it. And after that, I kind of leave it alone because I don't know enough about it as a teacher. They're there. Again, if you were going, you would learn more about those things and encounter them more. If you were working with healthcare, let's say Doctors Without Borders, you're on a medical. That's when you begin to become more acquainted. I know some of the students, I'm guessing any of you go to Ecuador this over Thanksgiving? No? I know a few of the students. But that's the kind when you start to become more familiar when you go to areas where you don't understand water supply. And viruses, it's something called prions. Viruses are not cellular. That's why they're not living things. Viruses are basically a chunk of either DNA or RNA, never both. Okay? With some protein around it, maybe some other stuff. Very simple. They're almost like, think of it almost like replicating poison. Idea. And indeed, Louis Pasteur, who we'll talk about, coined the term virus before he knew what it was. It actually did his aspects of his language and poison. He was fairly active. We'll talk a lot about Pasteur. And prions are actually misfolded proteins that have a bad habit of running into other proteins and causing them to misfold. And maybe the most famous disease that they cause is called mad cow. And oh, by the way, they're fatal. Just throwing that out there. And by the way, we don't have any treatment. So I mean, just little introductory stuff that are there. We'll get into all the ins and outs. Give you an idea. Here's that cell you're looking at. Here's our cell. Okay, maybe it's an epithelial cell. You can see how big it is. Okay. So take a look. Here's a eukaryotic cell. And now you can get, now we're looking at a bacteria. Okay, or a bacteria here. Some bacteria are shaped like rods. You'll learn about that in the lab. We'll talk about it as well. Some are shaped like little spheres. They're called cocci, hence the term staphylococci. Staphylo means a cluster, like a cluster of grapes that you buy at the grocery store. Cocci. Strepto, a chain of cocci. Very, very similar in size. E. coli would look a little bit like that. And now you get down here to some viruses. This would be a pretty standard sized virus. That'd be like a cold virus. This would be an oddball virus that affects bacteria. And then we're down here to just proteins. And you see the difference between cellular and acellular. The beauty of microbes and why we use them. They are how when we start to study diseases and medications and treatments, particularly medications, the first thing we study them are in the laboratory on bacteria. They're just like us. They're living things like us. They're not identical, but we're living things too. Like living things have DNA and RNA. Living things are all made out of cells, right? That's cell theory. They all have semi-permeable membranes. They all react to stimuli. So, I mean, those are basic things. They replicate. They do energy transfer. Kind of an easy primer about what they are. The big thing is DNA and RNA. So I was talking about why do we test things about watching E. coli growing because it's so easy to come across and grow. There's hardly any need to grow. You have a little water with some salt and some sugar, and you can grow E. coli very, very easily. And then we can study its effects because it's just like us. We have DNA, we have RNA, we make proteins with them, etc., etc. We're made out of cells. They're all cells. In fact, they're single cells, different. They all have membranes like we do. They react to certain stimuli or chemicals in their environment. They replicate, as you'll find, different than the way we would replicate them. Okay. And they do energy transformations, metabolism. Ingest stuff like we do, organic chemicals like we do. They generate energy and building blocks from them. They get rid of waste products. They're very much like us, except when you want to study something, you need big numbers. So you can think why we use them. They grow so rapidly. When we get to the growth curve, in a matter of, you'll see it in lab, in a matter of a day, you're going to say one group of bacteria can form millions of bacteria in a day. They replicate about every 20 minutes. They keep dividing in two. It's exponential growth. It's one of those things, like the growth curve, they go like that. We'll look at the growth curve. So we can get vast numbers. That's a big population to study. We can also, if it hurts the bacteria, it's likely to hurt us. Indeed, every chemical that we use, every kind of agent that we use has to pass a test we're going to learn about called the AIMS. Proper name, capital AMES test. You literally take that and test it on a bacteria to see if we can create a bacterial mutation. Very specific. And a bacteria that you've heard of called salmonella, a strain of salmonella. If it's able to mutate that salmonella in a specific way, we can't use it on us because it's almost surely going to cause cancer. So, I mean, there's so much with it. We have large numbers. They grow very, very rapidly. We can study them. I mean, you couldn't possibly get millions of people to do a study, nor would you, until you knew that the medicine or the agent was safe for them to use. There's the legalities of it are dramatic. So we use them. They're easy to study because they grow so rapidly. We can find them, we can identify them, etc. We can grow them in the lab easy. You're going to be using those little petri dishes, little culture plates. You're going to do the same thing. You're going to have millions on your plate right away. You're going to be able to identify them. You're going to be able to test them and study them in a variety of different ways. We can't see them directly, so that's where staining comes in, using the microscope. When we analyze them indirectly, we use a lot of what you're going to do is study what they make or how they use something. You're going to see, and we're going to talk about that a little bit, about fermenting sugars. You do more of that in the lab, but believe me, when you're growing stuff in that incubator in the lab, it's not going to smell great. It'll smell like stuff spoiling in your refrigerator, in my refrigerator. Effectively, that's what's going on there. And they undergo a process called fermentation. Fermentation doesn't smell good. When it's controlled, it does pretty well. It's like making wine. or making, alcoholic beverage. And obviously we use microscopes for it. Which of the following is not considered a microbe? A mushroom you can see with the naked eye. It's in the fungus family, but it's what we call macroscopic, which means with the naked eye. Fungi are big players in the microscopic. We have problems with them all the time. One of the most common infections people see and treat are yeast infections, particularly vaginal yeast infections. They occur all the time. Like I was on immunosuppressive drugs when they were actively treating aspects of my bladder cancer, which thankfully is not there right now. And I ended up getting thrush, which is an infection in your mouth with the same stuff that makes vaginal yeast infections called Candida albicans, which we'll learn about that. So I mean, I had to like dissolve throat lozenges 5 times a day in my mouth to get rid of it. Just hey. So understand that there's aspects of that are very important, and indeed, it's the way that they finally when the first paper was written about a disease called AIDS and a study of a virus called human immunodeficiency virus, we call HIV. It was identified in San Francisco because a cluster of gay male patients, rather large cluster in San Francisco, developed a specific and very, very rare fungal infection in their lungs, a fungal pneumonia, which your body only gets if you're very, very sick, immunosuppressed. This doctor assembled this and did a study and was able to identify the research in 1981, and that's when we came up with the name AIDS, acquired immune deficiency syndrome, and identified a particular virus called human immunodeficiency virus. And this now we know it's type 1 that was there. It was around before that. I'll give you a lot of information about that. 50 years ago, it was everywhere where I was training, and I did my medical school and residency in the city of Philadelphia, because the 2 largest gay male populations in the country were, percentage-wise, were in San Francisco and Philadelphia, by far. We were seeing this, and I'll give you a lot of background information about that. That's the advantage of having some old guy teaching this stuff. Sorry. No, I wasn't there working with Pasteur in 1860. Despite what you're all thinking. It's okay. You heard all this stuff before, I know. There may be new people here who haven't had me yet. They haven't heard all these jokes. I know, at least 2 of them. You're expected to laugh. Okay, microbes have shaped the development of the Earth's habitat. Single-celled organisms appear on this planet. Okay. That's based on carbon dating. I'm not an evolutionary biologist. I don't know anything about it. We'll go with it. It's science. Okay, I'm never going to ask you a question. How old is the planet? I don't know. Okay. So that's where the eukaryote, EU implies true. Karyon or karyo in this is the core or the kernel of a cell. So I use the term, and this is important going forward. Test questions are going to be phrased in eukaryotes or in prokaryotes. But when you're going through a question, and yes, you can write in a test, you're going to highlight, it says eukaryote or prokaryote. That's how you answer the question. Most important thing. Does it have mitochondria or not? Does it have membrane-bound organelles or not? We're going to go over it, but that's the important part. That's part of something you weren't particularly good at, which was test taking. I don't mean to be critical. I'm meaning for you. My job is to make you better at this, as well as teach you something. So true nucleus. Bacteria are called bacteria and archaea are called prokaryotes because pro or pre, the implication is before, pre-nucleus. They have DNA and they have RNA, but you're going to see it's not contained in the membrane. The whole difference, the whole show between eukarya and prokarya that are appropriately used, eukarya have membrane-bound organelles, including the nucleus. where prokaryotes do not. They do not have membrane-bound organelles. They have plenty of organelles. They're just not membrane-bound. It's a much less complicated cell. It does one thing very, very well. It reproduces. That's the big problem. I told you, why in 2 days all of a sudden do you have a massive bacterial infection in somebody? Because all of a sudden you're going to have millions, if not billions, of bacteria once they start to colonize and grow. And so you do something to stop it. If your immune system can't handle it, it's all part of it. Archaea, same idea. Single cell, no true nucleus. We'll go. I have a handout with all this. I'll go over it. I know there's a lot of information in a relatively short period of time that I'm giving you. This is kind of whoever that common ancestor. And there's a lot of names that are associated. These are the domains. Remember the domain system? Domain system was put together by a scientist named Weiss. Came out in 1978. It was based on ribosomal RNA and DNA sequences, little DNA segments. So he had done this when it was published. Obviously, the work was being done before. Up until that time, I got my doctorate in 1977. We were old-fashioned. We did kingdoms, animal and plant kingdoms. So a lot of us, people thought because bacteria all have a cell wall like plants do, they were microscopic plant form. So we called them microflora. Remember, fauna for animals, flora for plants. And indeed, you're going to hear the term in a hospital for somebody, you get somebody old like me, oh, that's normal flora. Meaning that, we got a culture back with this. Oh, don't worry about it. It's normal flora. That's normally supposed to be there. Even though it's not flowers or plants, they are bacteria, but that's terminology is something you'll learn if you get into a hospital floor or clinical setting. It's what we learned in school that we use for most of our careers. And so that's not an uncommon term at all. So Weiss back in 78 came out with this. We didn't learn that. I was there before the study came out. And this was the onset of microbiology or molecular biology. He studied ribosomal RNA and DNA sequences and found a couple of interesting things. Bacteria and archaea were somewhat different in the sense. Sorry, about 10 minutes. And you're hanging on everything. Sorry. Bacteria and archaea. Never mind. Bacteria and archaea. They're single cell. Have a cell wall. Plant, only single cells. Just like they have DNA and RNA. You're going to see that the DNA and RNA, even though they're similar, there are differences from ours. They're different, albeit similar. Not the same. The RNA specifically, you have to go with it. It's classified as something called 70S. S is a measurement unit called the Svedberg unit. I just go with S. It's easy. Learn the proper name. It has to do with density. They measure them and they spin them down and then centrifuge to figure out the density. RNA is constructed differently. The DNA sequences are similar but different. The DNA, what's our DNA? Our cells. 23andMe, Ancestry.com. DNA is found in what? It's in the nucleus. How is it configured? It's one giant strand. Pardon? It's a double helix. I'll buy that. Good, perfect. Pairs. One from mom, one from dad. Right? 23 pairs. XX, XY, that type of stuff. Okay. There are 23 pairs of chromosomes, okay? So they have pairs. These in bacteria and archaea, they have circles. DNA. They're large, big circles. Circles of DNA. And we in eukaryotic cells have for single or multi-cell. You can have just a single cell like a protozoan or multi-cell. Cell wall or not. You don't have to have a cell wall. Okay, they have both DNA and RNA. Here they have pairs of DNA that are linear. It's different. And the ribosomes, the RNA, has a different density. It's called 80. Those are the big differences between bacteria and archaea. The oddball about archaea is that the environment lives only in extreme environments. For most of us, we become very hot. That's what we did. We studied that and we created that table. Because they're interestingly, point I'm finally getting to. Interestingly, these guys, weird. Their DNA was a little closer to ours, little fragments of DNA. So we probably evolved, ourselves probably were more from this side of things and eventually leading to here. But it's very interesting, this classification, and we use it all the time. Oh, we got more time. Use it all the time. Big problem, the big thing with bacteria, they'll have a cell wall. Down the road, you're going to see that cell wall. And all this is going to go over again and that's working. There's a lot of improvement. In bacteria, every cell wall has this substance called glycine. EPTIP, you're going to be sick and tired here. Amino acids and sugars, ketoglycan. That's the big difference between bacteria and our cells. So I just said, I name a bacteria, and I said the bacteria, if you have a bacterial infection, what do you get? If you have a bacterial infection, what medicines do you get?
Antibiotic.
You do get an antibiotic. What would be the name of it? Big common antibiotic. Amoxicillin, very good. Amoxicillin is in the family of penicillin. Penicillins target the cell wall. Guess what they target? Ketoglycan. You pick something that our cells don't have. That's why the saddest words you can hear as a clinician are allergic to penicillin. It takes away your best category of antibiotics. What if you're allergic to penicillin? What do you take? That would be one particularly good answer. That's in the tetracycline family. What else? Famous because you only have to take like 5. Another antibiotic? Z-Pack? Doxycycline. Very good answer. Doxycycline targets the ribosome. Targets here. 70S versus 80S. That's particularly targets the 70S ribosome. So when you're allergic to penicillin, we'll take it just on something else. Typically, that category of antibiotics targets the difference between the ribosomes. That's the good news. There's bad news. There's more side effects. Because inside of our RNA, we're going to get to this. Inside of our mitochondria, our cells, believe it or not, they have some 70S ribosomes. So there's a whole lot of nuances here. That's the important part about this. Like, why do we have to study this and look at all the differences between our cells, bacteria cells, archaeal cells, viruses, etc., etc.? Because by understanding that, You look for the differences anatomically and physiology, and how do we treat them? We exploit them. This is different. Cell wall. I don't have a cell wall. Attack it. I have very, very few 70S ribosomes. Attack them. So that's part of that's why we spend so much time. So it helps you to be familiar. No one's going to ask you this stuff, but you're going to understand. Oh, why did I get this antibiotic? A patient might ask you that. I usually get a Z pack, or I usually get doxycycline. Why am I getting this today? And you'll be able to say, look, understand this is a different kind of infection, and it's got. We know there's a lot of resistance to that, so we have to use something special. Or this is a mutation. We don't have to use something; it's just different. Understand? And understand. I'll say this again and again. When a patient asks you a question, there's a very good answer, and a lot of times, well, I don't know. Don't BS. Goodbye. Have a wonderful day. See everybody Friday. Oh, it's just grand to be back.